We have seen that there is a great variation within various species in the nature. A new progeny is formed through the process of reproduction. Except for a few minor differences, the offspring show great similarities. This transfer of characteristics from the parents to the offsprings is called as heredity. In the ninth standard, you have studied about the DNA and the RNA. Though there are many similarities between the parents and the offspring, there are some differences too. These similarities and differences are all the effects of heredity.
Standard 10, Science and Technology Part 2, Chapter number 1, Heredity and Evolution. Introduction. Can you recall 1. Which component of the cellular nucleus of living organisms carries hereditary character? Answer. DNA carries hereditary character of living organisms. 2. What do we call the process of transfer of physical and mental characters from parents to the progeny? Answer. The process of transfer of physical and mental characters from parents to the progeny is called inheritance or heredity. 3. Which are the components of the DNA molecule? Answer. The components of the DNA molecule are two helical strands consisting of deoxyribose sugar, phosphoric acid, and pairs of nitrogenous bases. Heredity and hereditary changes. You know that heredity is the transfer of biological characters from one generation to another via genes. Johann Gregor Mendel is pioneer of the modern genetics. Around the period of 1886, it took a long time for him to understand the conclusions of his research about heredity. In 1901, the reasons behind the sudden changes were understood due to the mutational theory of Hugo de Ries. Meanwhile, in 1902, Walter Sutton observed the paired chromosomes in the cells of grasshopper. Until then, it was not known to anyone. Research started in the direction of finding the nature of genetic material when it was proved that genes are carried via chromosomes. Through which 1944 trio of scientists Oswald Avery, Macklin McCarthy, and Colin MacLeod proved that except viruses, all living organisms have DNA as genetic material. In 1961, the French geneticists Francois Jacob and Jacques Monod proposed a model for process of protein synthesis with the help of DNA in bacterial cells. It helped to uncover the genetic codes hidden in DNA. Thereby, the technique of recombinant DNA technology emerged, which has vast scope in the field of genetic engineering. The science of heredity is useful for diagnosis, treatment, and prevention of hereditary disorders, production of hybrid varieties of animals and plants, and in industrial processes in which microbes are used. Transcription, translation, and translocation. Can you tell? Sketch and explain the structure of DNA and various types of RNA. Answer 1. DNA. 1. DNA molecule is a double helix consisting of two strands. Each strand of this helix is made up of nucleotides. 2. Each nucleotide is made up of A. Phosphoric acid, B. A deoxyribose sugar. C. A nitrogenous base. 3. Nitrogenous base are of two types, viz. Purines and pyrimidines. 4. The purines are of two types, viz. Adenine and guanine, and pyrimidines are of two types, viz. Cytosine and thymine. 5. The adenine always pairs with thymine with double hydrogen bonds. 6. While cytosine always pairs with guanine with triple hydrogen bonds. 7. The helices remain bound due to these hydrogen bonds. 2. RNA. 1. RNA is nucleic acid having single strand of ribonucleotides. 2. Each ribonucleotide is made up of A. Ribose sugar, B. Phosphate molecules, and C. A nitrogenous base. 3. Adenine, guanine, cytosine, and uracil are the nitrogenous base substances which are present in RNA. 4. Adenine and guanine are two purines and cytosine and uracil are two pyrimidines. 5. RNA is found both in nucleus as well as in cytoplasm. 6. According to the functions, RNA is of the following three main types. A. mRNA. mRNA carries the information for protein synthesis from genes on DNA chain in nucleus to the ribosomes in the cytoplasm. Therefore, it is called messenger RNA. B. rRNA. rRNA is the component of the ribosomes. It helps in protein synthesis. C. tRNA. tRNA is present in the cytoplasm. According to the message of the RNA, it carries the specific amino acid up to the ribosomes as per the message coded on mRNA. Explain the meaning of genetic disorders and give names of some disorders. Answer. Genetic disorders are caused due to abnormalities in chromosomes and mutations in genes. Reasons of genetic disorders are as follows. 1. Increase or decrease in number of chromosomes causing numerical change. 2. Deletion of any part of the chromosome. 3. Translocation of chromosomes. 4. Sudden change or mutation occurring in a normal gene, turning it into a defective gene. 5. Mutations in more than one gene at the same time causing polygenic disorders. Examples. 1. Disorders due to numerical changes in the chromosomes. Down's syndrome, Turner's syndrome, Klinefelter's syndrome. 2. Monogenic disorders caused due to mutations. Hutchinson disease, Tay-Sachs disease, galactosemia, phenylketonuria, sickle cell anemia, cystic fibrosis, albinism, hemophilia, night blindness, etc. 3. Polygenic disorders, cleft lip, cleft palate, constricted stomach, spina bifida, a defect of the spinal cord, etc. are polygenic disorders. Diabetes, blood pressure, heart disorders, asthma, obesity are also polygenic disorders. With the help of RNA, the genes present in the form of DNA participate in the functioning of cell and thereby control the structure and functioning of the body. Information about protein synthesis is stored in the DNA, and synthesis of appropriate proteins as per requirement is necessary for body. These proteins are synthesized by DNA through the RNA. This is called as central dogma. mRNA is produced as per the sequence of nucleotides of DNA. Only one of the two strands of DNA is used in this process. The sequence of nucleotides in mRNA being produced is always complementary to the DNA strand used for synthesis. Besides, there is uracil in RNA instead of thymine of DNA. This process of RNA synthesis is called as transcription. The mRNA formed in nucleus comes in cytoplasm. It brings in the coded message from DNA. The message contains the codes for amino acids. The code for each amino acid consists of 3 nucleotides. It is called as triplet codon. Dr. Hargobind Khurana, a scientist of Indian origin, has made an important contribution in discovery of triplet codons for 20 amino acids. For this work, he has been awarded with the Nobel Prize in 1968 along with 2 other scientists. Each mRNA is made up of thousands of triplet codons. As per the message of mRNA, amino acids are supplied by the tRNA. For this purpose, tRNA has anticodon having complementary sequence to the codon on mRNA. This is called as translation. The amino acids brought in by tRNA are bonded together by peptide bonds with the help of rRNA. During this process, the ribosome keeps on moving from one end of mRNA to other end by the distance of one triplet codon. This is called as translocation. Such many chains come together to form complex proteins. These proteins control various functions in the body of living organisms and their appearance too. Living organisms can produce new individuals like themselves due to genes only, and some of those genes are transmitted to the next generation without any changes. Due to this, some of the characters of parents are transmitted to their offsprings. However, sometimes sudden changes occur in those genes. Sometimes any nucleotide of the gene changes its position that causes a minor change which is nothing but the mutation. Some mutations may be minor, but some may be considerable. Example, mutation may cause the genetic disorders like sickle cell anemia. This is an everlasting process and it is one of the proof for Darwin's theory.
Thank you.